Okay, I'd like to welcome people to our last April meeting. It's April 27th, meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. As always, I need to remind you that the meeting is live streamed and recorded, so please be aware of that. We're very glad that Claudio has agreed to come and talk to us about demoing their intriguing Jinko modeling platform. Looking forward to hearing about that. As always, Tom Helikar and myself are available to take comments, suggestions, Jim Sluka and Lorenzo as well. Don't hesitate to reach out to us if you need to tell us what we could do better or have suggestions for ways to make this work better. We have our usual communication channels. Please help us use them. In particular, we have the YouTube archive, the IMAG LinkedIn page, the IMAG Wiki page, and the Slack, and the Twitter. The Twitter in particular has a Twitter handle, MSM Viral. If you tag things with that, perhaps it helps us get the word out about what's being done here. So we appreciate your help. Many people will be moving to Paris for 2 or 3 weeks, in 2 or 3 weeks. The Building Immune Digital Twins workshop is organized by Anna and Reinhardt. Will start on the 15th of May. And I think they're pretty full, but if you want to go last minute, I'm sure they'll find a way of squeezing people in. That'll be very exciting. Look forward to that. There'll also be the IMAG MSM Consortium meeting, which hasn't happened for a few years because of COVID. It's the 20th anniversary of IMAG. That's going to be in person in Bethesda at the end of June. Registration deadline is May 1st, if you want to go to that. And then there's going to be, I think, a May 15th submission date for posters. Herbert Sauro is one of the organizers. Denise Kirshner is one of the organizers. Shane Pierce-Kotler is one of the organizers, and of course, Grace Peng. And there are going to be panels on a variety of things, including immune digital twins, organized by Gary Ann, so that could be interesting. And I do believe that they have this year, which is not the way it's always been, it's open to graduate students and postdocs. And they also have some funding available for grad student and postdoc participation. So you might want to look into that as well. I'm going to be going there because I was one of the, I'll be on a stage at part of it because I was one of the first people who got one of these IMAG grants when they created the program. And so they want the 4 or 5 of us who were original recipients to show up and sit there. I'm not sure what we'll do with that. I'll remind you that we are still doing this open rank professor search. It's really more of a senior search here at IU for 2 faculty positions in any area related to next generation biomedical digital twins. Could be everything from artificial intelligence approaches to data assimilation, to multi-scale integration, to mechanistic modeling, to experimental approaches to measuring health states with micro sensors. So if you're interested, please let me know. We have the GLIMPRINT website running. We can take membership fees. We're now officially a nonprofit. So if you're US, it's tax deductible to join. We'd love help making that work. So we look forward to hearing people's ideas about that. We have a steering committee meeting tomorrow at 11 a.m. Eastern Daylight Time. If you need a link, please ask us and we'll do our best to get you onto that. Are there any other announcements before we go forward? Okay. Coming up, we have David Basanta from Moffitt, who's going to be talking about modeling cancer ecosystems with agent-based models. Of course, He's done that both to do a lot of basic biology of cancer and also to do very nice clinical work to try to improve therapies, especially for metastatic prostate cancer. May 25th, we're not going to have a meeting because that's the middle of the Paris workshop. June 8th, we have Megan Hayes from University of Virginia, who's going to be talking about her multi-scale modeling of estrogen effects on muscle regeneration. And then June 22nd, we have Harm Huizingveld from the Netherlands, who's going to be talking about multi-scale modeling of toxicant-induced spinal tube defects. It's not exactly clear whether we'll take a bit of a break later in the summer. Depend a little bit on how many people want to present. Tom, do you have anything you want to add before we move forward?
I don't.
Okay. Well, I don't want to take any more time away from our speaker today. We're very glad that Claudio Monteiro is going to be showing us the Jinko modeling platform, a wonderful product developed at NovaDiscovery. And I will turn the share over to him.
Okay, so it's the second time I'm presenting the platform. So some of you had already a presentation of it. So there is Jinko. So it's a platform we are developing at NovaDiscovery. And the aim of this platform is basically to be first collaborative. So we develop it to help biomodelers at Nova to create models in a more efficient way, but also to have in the same place, different actors that can help or assess the different models and documents that we are creating. So, for example, the sponsors, external experts, or even persons from regulatory agencies can connect to the platform and see the different aspects of what is presented here, what are the main hypotheses, how we develop the models. So the aim today is to do a demo. So for that, I use a demo environment that we produced in that context. And we will have a presentation on NSCLC, the second version. So the version that is actually in development. And I will do the demo basically in the platform. So there is no slides. We'll use this platform to run all the demo. The aim is to have also the possibility to ask questions. So if you have any questions, don't hesitate. You can feel free to interrupt me and we can have discussions about that. So the model that I will use today is a model for non-small cell lung cancer. That is a subpart of lung cancer. And more particularly, a subpart of interest here is lung adenocarcinoma. That is also a subpart of non-small cell lung cancer. That covers, if I'm not wrong, almost 40% of all the lung cancers. That is leading cause for death in cancer in the world. And we have some specificities in lung cancer. Some patients have mutations in the epidermal growth factor, or EGFR, with different types of mutations leading to different types of targeted therapies that can be developed and had been developed in the past with different generations of mutations. And here we have a description of the model that changed a lot because we are working on that topic for more than 2 years now. We have a quite big team working on that. So we started with a small model, and then now we add a lot of submodels on that. So to give you an idea of the structure of the model, let's take a tumor. A tumor is represented with a n number of subclones, and each subclone has its own mutational burden. And we are doing that by duplicating the equations of each subclone and just changing in that context as the values of some parameters. And in each subclone, we have different submodels that are taking place. For example, here we have in dark blue the different submodels. So we have the growth factor signaling. We are particularly interested in that because we are working on EGFR mutant patients with different signalings in that. So with ARC, AKT phosphorylation, for example, we have the cell cycle and cell death pathways. We have the tumor growth that basically takes input the growth factor signaling and the cell cycle. And we have other types of connections with different other submodels like VEGF and the VEGFR. We have also, we are taking into account the death and the proliferation signals that lead to an increase of tumor growth in that context. Some phosphorylations that I mentioned before that are also an impact on tumor growth. And then at the end, for the primary tumor, we obtain a primary tumor size. And we connect this tumor size with the RECIST criteria. So basically, a criteria that is defined to clarify a tumor if there is progression or not, depending on the size of the tumor, but also if we have a different metastasis. And then we connect this tumor size, the tumor reduce, with one clinical outcome that is the time to progression. One thing that is to consider is during the first iteration of development of this model, we only took into account the primary tumor lung. And since a few months, we also duplicated this model in all the metastases that we are taking into account. So basically the brain metastasis, bone metastasis, liver metastasis, and the second lung metastasis. So the difference, so the tumor growth in both the primary tumor and in metastasis is taken into account with the TNM status and the time to progression. So that's it for the disease model. So basically the model that reproduces the course of a disease. And we will plug in this disease model different treatment models that have an impact on the mechanism of action. So for example, today we'll have a focus on EGFR TKIs that basically will have an impact in EGFR receptor. But we can also take into account right now in the model some chemotherapies with cisplatin, with pemetrexed. We can also take into account some immuno-oncology therapies with anti-PD-1. So this is not represented here, but it's also included. And anti-VEGF, for example. So that's the different connectors that we can use with different PK models to change the course of a disease. So that's it for this part. And as I said before, we are taking into account in the different tumors the heterogeneity by having different subclones. So for each subclone, we'll have a different types of response. And for each subclone, we can have the appearance of resistant mutations that will change the course of the evolution of the size of the tumor. And that's it for now for the context. I don't know if you have any questions until now, or if I can go for the second part with the demo. Okay, let's go for the demo. So two different topics to be presented today that we can explore. The first one is a study of gefetinib. So gefetinib is a first generation TKI, targeted therapy for patients with a mutation in EGFR, EGF receptor. And what I want to try here is to perform a dose escalation in advanced lung cancer patients. So I identified the paper, this one, where they are conducting this kind of trial. So basically what they are doing is they recruited a given number of patients, they enrolled them, and then selected 100 eligible patients that they randomized. And then to first arm, they will give a higher dose of gefetinib. And for the second arm, they will maintain the same dose and then see if there is an impact of using the double of the dose for months after 1 month of treatment. The conclusion here that we have here is there is no significant difference regarding the progression-free survival. That is not time to progression, but really close to time to progression. So we can use, for example, this thing, and overall survival for these two groups. So we'll try to reproduce this experiment and see if we have similar results with the model actually. And we have also some curves, maybe that we can use at the end to qualitatively compare the results. So the way we are doing it at Nova, it's always the same. So if we want to test that, I will start by creating an assertion. So let's take this one with the two groups. Okay, let's take the extract protocol. Okay, this way. Classification, I can put excellent because it's just a protocol in that context. And I will do the same for this figure. So I will take this image just to use it later. Okay. And here we go. So we'll go back to the document that I was presented before. I will add in this section the two elements that I created just before. So what I want to test is here. And the figure I want to compare the result is this one. So I will have to run a clinical trial with a total, that what they did is a total of 100 patients randomized 1-1. So what I will create basically is a virtual population of 50 patients because we run the same simulations in the same virtual patients. So in that case, we want to compare one by one what will be the results with a double of the dose. So to do that, I will create here a folder. With simulations, and then let's create a trial. Those escalation. Okay, let's select the computational model. So the model that we are using is this one, model V2. And here we go. So model is loaded. We have the duration of the simulation. By default is 2 years. I think we can stick to that in that context. And we have by default the outputs that will be rendered directly in the simulations. So we'll have the tumor 1 reduced, so the principal tumor, the time to progression, and the doses of getivetinib in that context. So to compare if we are really sure that we are doubling the dose. So to run a simulation, we need 3 different things. First thing is a computational model that is already connected with getivetinib. We'll need a virtual population, so to run the different model in different patients. And we'll need to create protocol arms to define what we want to run, what we want to simulate in that context. The measure design is just to define what we want to observe, what we have to analyze at the end of the simulations. And that's it. So we selected the model. Let's go for the virtual population, and let's create a virtual population in that context. So to create a virtual population, we'll create virtual patients, and a virtual patient is a vector of parameter values that are picked in distributions. So that's why the first thing when we are creating a virtual population is to define what is the model, but because the model will be used in that context. So let's create the dose escalation VBAP. And here we go. So by default, we have 249 descriptors in the virtual population. So each virtual patient will have 249 values that will be picked in all these distributions that's here. So as I said, I will generate a VPOP of 50 patients. Name. And we can select a seed in that context. So the seed is important if we want, for example, to use the same seed for a virtual population of 1,000 patients or 1 million patients. it will use the same seed as next time I will use it. So I will take the name seed 1 and let's go. And here we go. So we have generated 50 patients with values picked in all these distributions. And if you are interested, for example, we can click in the patient table and see what are the values, for example, for all of these patients. For example, patient 1 have 53 years and so on. So we can explore the different values for all these patients. And so basically when we run a simulation, for each patient, we'll be executing the model and see what will be the results. So let's go back to the trial design. Let's select the virtual population I just created. And let's start with the protocol harms. So we'll create a protocol harm. Here we go. So we want to compare gefetinib harm. Let's select descriptors. So what is interesting here is to have the doses of gefetinib. So what is administered to the virtual patient, if gefetinib is administered or not. And we have also in that context, the dose escalation. So it's directly a parameter that will define if we are applying a dose escalation in that harm or not. So we'll select the 3 different descriptors here. So for the first one, gefetinib, we'll just generate a harm where patients are give this drug with a normal dose to these patients. So first arm is gefetinib administered. Answer is yes. We'll duplicate this one, and then dose escalation. Gefetinib, and in that context, we'll compare it with the arm with gefetinib without dose escalation, and the answer is true. So we have 2 arms, one with gefetinib, where it is administered from T0 until the end of the simulation. And we have a second arm with the dose escalation, where gefetinib is administered until the end of the first month. And then we'll multiply the dose by 2 at the end of this own month until the end of the simulation. So maybe let's create a measure design in that context. So to have what we want to observe with the results. We'll select model output. So what we want here is tumor reduce. So we'll see the impact of the drug in tumor reduce. Let's take the tumor 2. So tumor 2 corresponds to the first metastasis that will appear during this kind of disease. So basically in the brain. And let's take also the time to progression. Here we go. So for the time to progression, what we need to take and observe is the time of max of this value. We'll call it TTP. And we have the tumor reduce. One, tumor reduce 2. And that's it. So we have the model, the virtual population, protocol harms are defined, and user design. So let's launch the trial. So when we launch a trial, we have a pop-up explaining the number of solving tasks that will be run in that context. So 2 arms for 50 patients, it's 100 solving tasks. And now we are running the model. And that's it. So when we launch a model in the platform, basically, we have some time that is the time needed to have the platform dispatching the different solving jobs. So we are using Amazon clusters to run the different simulations. So it will identify the number of clusters and the number of CPUs that will need to run the simulations, and then it will start. So basically, for each virtual CPU, if I'm not wrong, we have 2 threads in that context. So basically, in that context, we'll just generate 50 different jobs that will run. It can take like few minutes to dispatch the jobs, and then it will start, and we'll have an idea of how long it will take. So during the simulation, that will take probably like 4 minutes, 4 or 5 minutes. It's time if you have any questions.
So I got a quick question, Claudio. So the signaling model, is that an ODE model?
Yes.
Okay.
Yes, it is. Yeah, I think during the simulation, we can have a look to the model itself. So we have the computational model. So yeah, it's also one thing that we have in the platform. Everything is linked. So when you are running a simulation, so now I started the simulation, I cannot change the virtual population in that context. I cannot change the protocol arms because it's already running. So for need of traceability, we'll not be able to change, but we can, since it's launched, we can just change the page and go and see what is the composition of the computational model. So let's see it in a new tab. The computational model is huge, so it takes some time to oh, I don't know if you are seeing my screen. Yes, you are. Okay. So it takes some time to load. Basically, here we have some metrics, so we have an idea of the composition of the model. So we have 2,000 reactions, we have 300 ODEs, and 5,000 parameters in that context. So it gives you an idea of the size of the model that is running right now. And if you are interested in how we are implementing it, so here we have basically a documentation that is automatically generated from the code. So basically, we have a serialized model that can be, for example, in SBML format. And then automatically it takes the information that we have in the serialized model and it generates the documentation for you. So for example, if we want to have a look to the cell cycle, we have for the cell cycle, all the ODEs compose the cell cycle. So as I said, we are making a duplication for each tumor of the different equations that are happening in the different subplots. So that's why we have tumor 1 and we'll have tumor 2 and etc. And the model that I am presenting here is a reduced model. So it's basically used for the demo because it's more efficient. We are only having 2 duplications. The model that we are actually using to answer the scientific questions that are asked by the sponsors have 17 duplications for each tumor. So basically, we are taking into account also for metastases. So you have an idea of how much model is big when we are running it. And for example, for each of the equations, we have the documentation. So for example, here we have the cells in G1 phase. We have the equation. We have the dependencies of, for example, this equation with the parameter values, the unit, the distribution when it's part of the virtual population. So it's not the case here. We can also click on transitivity. So we will have appearing the parameters that are not directly linked with this equation, but are linked with some of the variables that are used in this equation. So it makes a lot of parameters in that context. So that's it for the basically the computational model and how it is represented. So for example, we have also for some of the parameters some conditions depending on if you are simulating on, for example, on human, or if you are simulating on mice, or if you are simulating in vitro settings, because the model has been developed using data from in vitro experiments from mice and human. So we have a translational model, basically.
And is each subclone, does that mean a different instance of the same model with different parameters? Is that what you mean by a subclone?
Yeah, exactly. So that's why, for example, if you take a tumor something very heterogeneous, that is basically supposedly the fact in real life. That's why we have, for example, some treatments that are really effective at the beginning of a simulation, because they are really impacting some subclones that are really sensitive to this treatment. And then you can have a sort of equilibrium of the effects. And if some resistant mutations can appear in the different subclones that are not responding to the treatment, then you have the tumor size that is increasing again, and the cancer that is progressing again.
So how do you simulate the appearance of mutations? Because that would generate a new branch of a new instance of your cell type model, right?
Yes.
You have an event that then clones the model, changes parameters, and creates a new submodel.
Yes, this is exactly that. We have an event in that context, and to calibrate the proportion of these events, we use some database that have been generated in the past by some teams. So basically, we have a seminal work done by Ajani et al. I don't remember exactly the date, but they took basically a lot of samples from tumors, and they explore the different mutations, including the driver mutations and the mutations that appear with time. So this gives us some ideas how the proportion is of appearance for some resistant mutations depending on the context. And then we created this way to develop and to take into account the appearance of resistant mutations.
Could you just show us in the code how events are handled? In other words, how does the model spawn another submodel, for example? How is that specified in the model?
Yes, this is a very good question. I don't know if it is feasible to observe it directly here. Let's check. Normally, that depends on the type of equations.
Because that tends to be a problem with SBML, right? SBML wasn't designed to have more than one instance of the model, and so you have to actually copy the text file and then overwrite the text file. It's so getting that to work with that kind of model specification isn't so easy.
So, for example, here we have no event for this equation. So let me check if I can identify one. It will probably not be in this model. It will be in EGFR, for example. Let me take, yes, this one, or let's take a different one. Let's take maybe this one. Depends on, no events on this one. Let me have a look to EGFR. This model is so huge, so it's a bit laggy. Okay, so I have seen. Okay, I'm not seeing any events here. I will try a new one, and if not, I will give you, basically, I will send you maybe that as a follow-up, just to give you an idea. But basically, an event is something taking this value at some point during the simulations. GFR. So yes, this model is too big to be reached directly like that. So we have units here. Let me check if we can just see. Maybe it's something that can be explored later with the second thing. Let me maybe continue with the simulation, and I will try to explore it with different documentation afterwards.
I'm sorry, I didn't mean to derail you, but that's a specific interest of how you implement mutations in an ODE framework is non-trivial. And so
yeah,
I'd love to see more about that when you have a chance. But thank you.
Yeah, sure, no problems. I will try to show it later, basically after that. So we have the solving that is completed right now. And we were supposed to have 100 patients, and we see that 2 patients failed, basically. So it's interesting because we can also explore why we try to have sundials, that is what we're using to solve the models, a bit more verbose in that context. So we identify that in the muscles, for example, gefetinib had a value that is infinite, so probably a value very close to 0, and solving issue with 2 different patients. Probably we're exploring values a bit too large in that context. So not a problem. So we'll just have these results happening with, so post-processing is ongoing in that context. So we have some time maybe to explore.
So I don't know if you're showing a different screen, but we're stuck on the list of parameters.
Sorry.
There we go. Okay, no worries. Thank you.
So yes, I will say again. So we have 100 patients that are being simulated. So we have the post-processing that is ongoing. And we see that we have 2 different patients that are in red. So this happens sometimes with simulation errors. And basically here is because in some variables, we have a value none. So basically it's probably because we have a negative value or division by 0 somewhere. So it explains that this happens in muscle and in veins. So probably some values that are extreme values in the parameters. So yes, since it's actually post-processing, let me check if I can see some documentation here on the knowledge. Take some documents and let's take tumor mutational profile. And let me try to answer your question. So this is particularly of interest because we have the different computational models. Tumor growth. So normally,
okay, so we're also not resharing the screen. We're seeing the outcome of the simulations that you.
Okay. And now?
I see it.
Okay. 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 So let me try to identify how we take into account these events. Maybe if we have these computational models directly, no, it's probably here. Let me check if I can identify. But yeah, let's use NSCLC V1 because it will be definitely smaller in that context. Cell proliferation. let's take, for example, this one. 7 events. So perfect. So you are all seeing my screen. So for example, we have in the tumor clone number 10, living tumoral cells that are going to 0 at time 0. So that is the kind of event that you can have. And it depends on tumor clone 10, for example, in that context. We have tumor clone 1 or tumor clone 10 if it's removing. So this is an example of how the events are happening in that context to remove the different tumor clones. So, and these tumor clones are removed by immune system in that context. So when we have one value that is reached in that context, so the tumor cells are removed by the immune system. So this is one example. Let me check if I have different ones. Immune recruitment to tumor site. We have this one, for example, and we have one event in that context. So the tumor cells go to 0, and the condition is the tumoral cells inferior to 1, and it depending on the count of cells and the number of living tumoral cells. And that's it. It's one other example of what you can have. And we are using quite a lot in that context, but it depends on the way it's implemented, for example. Here again, we have 7 different events. So yes, so can probably have also.
So this is, so well, I guess maybe there were 2 different kinds of event questions. One of which was the one you said, which is at time X, something happens to a parameter value. But if you have a mutation, potentially you're cloning a new model, because you have the existing cells that have some particular model, and now you create a copy of that model, a template off that model, which has different parameter values, because they'll run together, because the tumor will have both cell types.
Yes.
So I guess I was wondering a little bit about how you actually do that. In other words, is there a command that says essentially clone model or something of that kind that during execution as the thing is executing?
It's basically appearing during the simulation. So when some conditions that are dynamical conditions are achieved, then you have this event that will change the value, for example, of a parameter. So this will update basically the parameter value in that context, and then the simulation is going up again. So it's perfect. I don't know why this is stuck in that context. Try to resume it again. I don't know why the post-processing is so long. I tested just before. It's supposed to be definitely more reactive. Okay, solving completed. Phew. So let's go for trial visualization. So we have the dose escalation, and here we go. So we have different ways to observe the results. We have at first the scalar results. So as I mentioned, I defined the measure design at the beginning. So we are seeing my screen, right, with the simulations. Okay, great. So I defined 3 different measures at the beginning. So we have the time to progression. So basically it's a time between the administration of a drug and the first progression observed in that context. So the lower is the worst case in that context. We have the tumor radius in the different tumors. The tumor radius 1 corresponds to the primary tumor, and the tumor radius 2 corresponds to the secondary tumor, so the metastasis. So as mentioned in the paper, they mentioned that there is no major difference between both arms by doubling the dose after 1 month. So it's basically what we are observing here in this first thing. So we have a view of the population, basically. In the different data points here are the different virtual patients with the different values of TTP. And here we have just a box plot of the same results with the distribution. We can go now to the time series. So in that context, we have the evolution of the tumor, different tumor radius, and with the 2 different arms. So as a reminder, we have a first arm with gefitinib that is administered at T0 until the end of the simulation. And we have a second arm with a dose escalation. So we have the same dose until the first month. And then after the first month, we are doubling the dose. So we can observe that basically with the doses of gefitinib here. So that's the results. And here we see exactly that. So we have the dose of gefitinib that is 250 milligrams at T0. And then after, let me maybe change the time scale. I'll take 1 month. There we go. So after 1 month, we have the doubling the dose for the arm in blue in that context. So what you can see here is basically that we have no major difference between the 2 arms and the populations. So in the principal line is the median value. We have the 25 quantiles here in dashed lanes, and here we have the 2.5 and 97.5 quantiles. A different way to observe these results are the survival analysis. So in that context, we need to select a time to event. So we have the time to progression. And then we have a different way to observe these results too. So we have a Kaplan-Meier-like curve, and we see that in both arms, the results are quite similar in that context. So we can also, in that context, change time scale. We'll take it in month. If I'm not wrong, I think it was in month in the paper I identified. And we have also what is interesting here, a comparison automatically statistical tests that are used to compare the different arms. And we see here that according to the p-value, there is no major difference between these 2 arms, basically. So we can do 2 things. First thing is to use this result as an item and put it in the document directly, or download the plot as a PNG. So I will basically download this plot and put it in the document I started to work with at the beginning here. So let's go back to that. Let's go to simulations. Here I have this document. So I will add directly this figure here. And we see in this figure that basically, let me just remove it. It's not at the right place. So if we have a look to the 20, okay, that's not perfect in that context, but maybe let's include it directly here. And basically what we have at 20 month is at, let's say, 0.02. That is probably something that is similar that we have here to the different things. Let me go back to the simulations. And see if you go back to these results, see if we can zoom and maybe have a better. Okay. Let's load this one in the document. Okay, definitely better in that context. So yes, we are around 0.2 for the both. So something quite similar that we have here, basically with the simulations. So that's for the first thing, basically comparing those escalation effects that we can have in a group. Something that is quite important here is also that we have the progression-free survival in the first one, and we have the TTP in the second one. So not exactly the same metrics, but are comparable in that context, I think. So that's for the first example that we can use the platform. Maybe one thing before to start with the second one. The second one is basically what will be the difference in terms of efficacy in a given population of two different treatments. So a first generation and a third generation. And basically the difference that is between these two populations is linked with the appearance of resistance mutations. So what we can do also when we have this kind of results, if we go back to the simulation they presented just before, one thing is a filtering. So we can include eligibility criteria in that context and explore basically some characteristics of some different patients that we have here. So for that, we need to create a filter and the quantity of interest. And for example, let's explore if in these 50 patients, I don't know if it will be the case, but let's explore that, if they have a resistant mutation. So one of the resistance mutations that are actually impacting the efficacy of gefitinib is the T79M. So let's start a different way, is EGFR T79M mutated subclone or tumor, it will be even better. So let's take this operator like that. And yes, we have apparently one patient in the 50 that have this mutation. It is a resistance mutation. So now I created it. Let's explore which patient. So we have only one patient. It's a patient number 13 that have this mutation. So let's select it. So by doing that, I will highlight this patient in a virtual cohort that I created just before. So if I go back here in the simulation, the patient will appear to clean the results. So here we go. So we see that this patient 13 is quite in the top in that context. So the treatment has quite no effect in reducing the tumor radius. So this is part of the patients that are typically resistant to this treatment. And we should propose to these patients a different treatment in that context. So that's why we will explore basically with the second use case. So if we go back to, oh, my bad. Let's go back to, sorry, are you seeing my screen? I think yes. So let's go back to this testing example with the second one, and I will open this paper that is basically a paper from a clinical trial, quite well known. That is a Flora, if I'm not wrong. And in this trial, they compare the efficacy of two generations of EGFR TKI. We have gefitinib, or if I'm not wrong, we have a second one that is erlotinib. So two first generation TKIs, and they are comparing the efficacy of these TKIs with osimertinib, that is a third generation. So basically a a drug that has an impact on the mutation that I explored just before in the cohort, the EGFR T79M resistant mutation. So I will proceed in the same way that I did before. So let's create basically here an assertion. Okay, so let's go back here to text. Let's put here just here, and I just forget to take also a picture of one of the image to compare the results at the end. So let's do it again. Let's export one of the figures. And let's take this one, that is a full analysis set. So they are comparing all the population that they are using. And let's select only this one. There we go. So we have in blue osimertinib in this population. And in red, the first generation TKIs. So unfortunately, we don't have access to the drug model of erlotinib, so we'll assume that both gefetinib and erlotinib have the same effect in the population, to be in position to compare it. Okay, let's take this like that. And here we go. Let's go back to the document. Let's drag and drop again this image to be used. Okay, and then let's rerun a simulation and compare the results. Create new trial simulation. Let's name it like that. We select the model, we validate, and we go back to this same thing. So let's create a virtual population, maybe this one, a population a little bit bigger. There we go. Size of 100 and use the seed number 32. Okay. Let's select this population that is appearing right here. And then let's create protocol arms. So what we want to do here is to compare two arms and to use this two arms comparison to compare the efficacy of the drug. So I will start with, maybe let's start with a placebo arm, so something that we can do in real life. So natural history. So basically a arm that is, we are not giving any drug. And then we want to test gefetinib. So this gefetinib human is gefetinib administered. We want osimertinib. Osimertinib again is administered. And what I want to test in this simulation that is not possible in real life is, let's use, for example, here an event and add in some arms the fact that all the patients have this resistance mutation. So is EGFR because it's in protocol specific, so is EGFR mutated tumor. So we'll add that also. So first arm is natural history, so doses will be 0 for both drugs. Is EGFR mutated, we'll put it in 0 in that context, so we'll not override the values from the virtual population in that context. And then we'll create additional arms. So first arm with gefetinib. Standard dose is 150 milligrams. If we overlay, I don't know if you see the bubble that appears when we are overlaying. Yes, it is the case. So this is a dose in, no, we are not seeing that. Okay, so in my screen is appearing, so a sort of little pop-up with a definition of what is the descriptor. So we are comparing gefetinib with the natural history in that context. And is gefetinib administered? Answer is E1 for yes, of course. So we'll create a new arm that will be named osimertinib. But it will be compared to natural history in that case. Is EGFR mutated? Answer is no. So treatment doses for gefetinib is 0. Is osimertinib administered? Answer is yes, 1. And I think it's okay for this one. I think we are missing one of the descriptors, that is osimertinib dose. Let's use again protocol specific to select less parameters. Osimertinib. So I think I included, oh, it's included, but not here directly. Okay. So maybe let's change the way it is displayed. Okay. Just not, okay, there is something interesting appearing here. Let me reload the page. I think there is a glitch. Okay. Okay. So yes, one was missing. Osimer. Okay. And now we have the dose. So natural history, the dose will be 0. Gefetinib arm, the dose will be 0 in that context. We have osimertinib arm, the dose will be 80 milligrams. And that is the dose by default, and it is administered. And then we'll just duplicate these 2 arms, and we'll add specificities on that. First specificity is that we'll create C7090M mutation in all the patients of this arm. So is EGFR mutated tumor? Answer is yes. And we'll do the same thing for osimertinib. So here is yes. Okay. So normally, let me check I'm not wrong. Natural history here. We have gefetinib administered with the standard dose, gefetinib with the mutation administered and the mutation, osimertinib standard dose, and osimertinib with the mutation. So normally we are okay. Let's create a measure design. What we want to observe is again the same thing. So we want to observe the time to progression. We want to observe tumor radius for tumor 1, so the principal tumor, and tumor 2. And for time to progression, we're interested in time of max. Okay, TTP. Tumor 1 radius, tumor 2 radius. And we can launch the trial. So 100 patients, 5 arms. And what is interesting here is normally the simulation, even if we are running more patients than in the previous trial that we simulated, this simulation will probably go faster than the first one, because the different virtual CPUs are already allocated in the platform. So it's like 15 minutes, and then it will decrease quite quickly. We are just assigning the different jobs. So do you have any questions during the simulations? I think we have some time for that.
I'm just incredibly impressed by the software. I'm taking all these notes about how we should really. I wish our software were integrated into a framework like this, so that
it could be doable. By the way, if you want to test the platform, we can create accounts for you with some models. So probably the version 2 we are working on right now will not be available, but I think we can give you access to the first version of the NSCLC model, so the one we are publishing actually.
Right.
So
I mean,
it's not the
specific, it's not the specific model that's important. It's understanding how the user interface works, how your control works, the ability to define the virtual population. There are all these. It's very elegant. It's really impressive. Yeah. So I don't really have a question except to say I'm extremely impressed.
Maybe the time that is running, if you want, we can explore some aspects of the virtual population. We'll be happy to do that. And maybe, or let's create a new one as time that is progressing the simulation. So normally, yes, it's becoming to be really fast now. We have 5 minutes left and probably 2 minutes in 5 seconds. So maybe let's have spend some time having a deep review of the virtual population, because I just generated a virtual population by default. But we can go further like that. So let's take a model, for example, here. Let's take this one. And as you can see here, one example is we can use a tool to generate a virtual population, or if you have your own virtual population. So basically, if you generated your own patients, then you can import your own virtual population in that context. So if you have a CSV with the same parameter names, you can decide to generate your own population and load it in the platform. It's doable. So here we go again. So we have the selected descriptors by default, but if you want to add more, it's totally doable. Basically, we can add all the parameters from the model if you want, but there is no interest in that context, because some parameters, maybe you don't want to make them value, but that's the thing that you can do. And then let's take, for example, the age. We have the age, and the age defined by this theoretical distribution. And if we want, we can change it. Here we have a normal truncated, but if we want, let's take, for example, a normal distribution and have a mean of, because we want to explore the effect of a drug on older patients in that context. So we can take, I don't know, 75 and have a ASD of 1. So here is the age distribution. So if you want to generate patients in that context, we'll generate patients in the distribution in particular. One thing that is also quite interesting is the correlations. It's something that I did not mention during the demo because I was just generating virtual populations by default. But if you want to make sure that a patient is realistic, we can take into account correlation observed in real life. For example, we know that maybe males will probably have a more bigger proportion of males with a smoking status of yes, we are smoking. It's part of maybe the what is observed in real life. So it's something that we can take into account in that context. So we can take the smoking status, is this dimensionless in a context, and is male, and we can add a coefficient. So basically 0.4. And here we go. And we can even add a reference. For example, if we have an idea in the real population, if we have studies explaining that, I don't know, we have more chances of smoking if you are a male, you can add the reference here, and it will be stored in the virtual population that you generate. And then in that context, you will be generating a virtual population that will take into account these different correlations, its pairwise correlations that we introduce at the beginning. Okay, so let's go back and see how the simulation is going. It was a trial simulation. So apparently it's finished. Yes. And let's create the results again. And the reason why we are each time creating the results, it's quite important, because when you create the results, you are creating also an item. And this item can then be used and be imported in a document. So when you are sharing your results with a colleague, for example, you are not only sharing plots results, we are basically showing that. We are showing this page, and we can, of course, observe the results, maybe add some filtering options, have a better view of, yes, what is happening in that context. So here we have the same thing again. So let's maybe use a box plot to observe the results. And okay, so we have the time to progression that is quite different. So we see that time to progression with lower values are here, time to progression with higher values are here, and the two time progressions with higher values are basically the ones with osimertinib. So if we go to this part, let's have a look to the tumor reduce. So we have a lot of arms here. Let's select only basically natural history. And yeah, so the natural history is we are not giving any treatment, so the tumor is progressing. If you had gefitinib in that context, so I probably mess up in the trial design, because gefitinib is not having any impact here. Okay, I think I changed the name of gefitinib and gefitinib 70M. So here we have the normal gefitinib that has an impact, and then we know that at 200 days approximately, we have the appearance of resistant mutations that lead to an increase again of the size of the tumor in the population. But some patients don't have the appearance of these resistant mutations because they are responding quite well. If we take osimertinib in that context, okay, I think it's this one again. There is probably a failure. I will have a look to the simulations afterwards. We see that osimertinib have quite a huge impact here in terms of resistance, and there is no sensitivity in the resistance with that. And probably here we have gefitinib with a resistant mutation, and here we have probably a mix of osimertinib. And if you have a look to the survival analysis and the time to progression, we have the different arms here. So we see that natural history is really close to the gefitinib with the mutation in that context. So there is no impact here in the results. And then you have the osimertinib arm that is a mix of different types of mutations. And we have the gefitinib as the osimertinib arm here, but only with patients with the mutation of interest. And this is typically an effect that we have because the population is, we know that there is an impact here of the mutation on the response to the treatment. I will just have a look here on the trial simulation, maybe, because it's up here a bit strange. Maybe I think I have an error in the name. So for gefitinib 70M, okay, yes, I mixed up in the terms. Sorry for that. That's why the results are a bit different here. But I mean, anyway, it doesn't change the observations we'll have here. So if you go back to the simulation, what we can observe here is the fact that we have this impact of this mutation in the results that we have in the simulation. One thing that is also interesting here to show, I did not show it before, but we have the contribution analysis. So the aim of the contribution analysis, maybe I will just only take the time to progression because it takes quite a few time to run. It will create tornado plots based on one output and see what are the more impactful parameters in given this output. So basically it's a sort of sensitivity analysis that we can perform and give you us an idea of what are the more impactful parameters in the simulations. So this takes some time. Here we go. So if we take, for example, let's take one harm in particular. Let's take, I don't know, this one. We see that parameters that have quite a huge impact in the answer to osimertinib's arm with the mutation is the fact that they have also the TP53 mutation in these patients. We have also the quantity of E2F, that is transcription factor impacting the cell cycle. We have also the fact that we have some resistance that are added to that. So we can explore afterwards basically what are the parameters that are more impactful. For example, if we take the gefitinib harm, interestingly, one of the more impactful parameters is the initial value for TNM. So TNM is a classification of the different grades of cancer for patients, with T the size of the principal tumor, and the fact that some patients have tumors in nodules, and M for metastasis. So basically, here we see that it's really impactful in the response of patients to this treatment. And yeah, that's one of the simulations that we can perform.
I think I'm afraid we're out of time for today. This was fabulous. I'm going to have to watch the video a couple of times to review it. It's really impressive. The user interface is great. The capabilities are great.
I'm happy to have this answer. I don't know if maybe if you have maybe some time to explore other things if you're interested in. Happy to show you other aspects. But yeah, that's almost it. We continue to work on this version of the model, and we are adding new treatments and comparing it with literature, with databases to see how predictive it is. We are actually in the revision of a paper that will be submitted to Nature Systems Biology and Applications. So basically afterwards, we'll have the first validation process of the NSCLC V1, so the previous version of the model. And we are working in the new version for more than 1 year now.
Any other comments or questions? Lorenzo, Jim? There's so much we saw. It's a pity we don't have. We'll have to have you come back and do it again, because an hour and a half isn't enough time.
Yeah, we'll be happy to present you again, or with a different disease model, if you're
That
interested.
would be fine, Jim. Lorenzo, go ahead.
No, nothing. I'm very impressed as well. It's really amazing. The whole workflow, it's very complex, and yeah, for sure it needs a little bit of time to take on all the things. I guess the ultimate point will be the model themselves, right? The underlying model, and to really see how we can make it in such a way that little by little we include all the information beside that is the most obvious, and then we will include rarer variants and mutation and things that make it even more and more predictive.
Mhm.
That's true. We didn't really drill down into model structure here. You showed us a few ODEs and things, but
Yeah.
that was, I think Lorenzo is right. I mean, I think the ability to build the model and the documentation together is very powerful.
So I can tease you a bit on that. So for now, we had to make some choices in the development of the platform, and instead of giving the ability to develop and edit some model directly in the platform, we prefer to basically use your own developed system, so basically code outside the platform, and then generate SBML files or serialized files for the models. And the ability to edit the model will arrive at the end of this summer, probably. So we'll have an alpha version that will be released probably last quarter this year. So if you are interested in, it will be also possible in the platform to start editing the models. So for now, what we have is a generation of the documentation based on a serialized model, so a XML, so a SBML file. Soon it will be possible also to edit the model in the platform, to change something in the model, maybe to test different equations, maybe to add some scorings or add some events, change the events. So it will be interesting to maybe fine-tune the model directly in the platform and see what will be the difference in terms of results. So this is coming, and it's basically we need to make some choices in the development, and yeah, it will be part of it. We have an example here that is quite interesting. It's what I was explaining just before. So when you are creating, for example, a computational model, protocol harms, it becomes items. So you can share these items with other people. So we can share virtual populations, and we can also share simulation results. So I don't know if this one is working, but let's test it. Hopefully it's still working. The simulation results for a different model. We imported, that's why I think this example is also interesting. We imported a model from literature, and this model was about neutropenia. So to give you a bit of context, when you are giving chemotherapies to patients, one of the side effects is neutropenia. So basically, this is destroying a lot of things, including immune cells. And you are monitoring the level of neutropenia for all these patients before to give them a second dose. And some industrials developed some drugs to boost the production of immune cells, and then to be able to give these treatments sooner than later to the patient, so they can benefit from it. And then we have an aspect that is very important, that is the timing. When should we give the chemotherapy? When should we give the booster in order to optimize the number of neutrophils in the blood that are circulating? And I'm afraid that this simulation is not working. Oh, yes, it works. Perfect. So we simulated different things in that context. We simulated arm with a placebo, so we are not giving any booster to these patients. And then we created their arm with, we are giving the booster too early, and we are giving the booster too late. And what we are observing here is a count of number of cells, of circulating cells in blood. And interestingly, we see that with the right timing, we are optimizing the level, so we can administrate the chemotherapy sooner than for the other patients. So basically, it's a different usage of models in the platform, and then we can test different regimens and different combinations. And it's quite interesting in that context. But one different examples. And this model, for information, it's really simple in the way it's implemented. That's why we tested with this one. Let me check if we have this model here. We have the computational model. So it loads so fast compared to the NSCLC model. So we have 10 ODEs and 72 parameters, so 10 variables. And we have 3 events in that context. And for example, we have the blood receptor that depends on that. So these equations are quite simple in that context. So it's really easy to explore that. And we have, for example, for this parameter, we know that if this is used in the virtual population, and we have here, for example, the distribution. It's a uniform law with the different values. And I don't know if we have, for example, this one. This one, we have an event. And we have this event that is here. That's the CRT with the doses of corticosteroids and with a lag that we introduce. And that's it. It's one other example that we have. So this is a demo environment, basically. And if you are interested, when we create accounts, we also create a sandbox with some models that we can explore. Or if we have models in SBML format, we can also help you to load this model in your own account if you want to test it. Sometimes it's easier to use our own models because we know them, and it's just a different way. It's a good way to compare basically what we are used to test them.